Right. Hello, I'm John Black, and I chair a group called the Pharmacogenomics Task Force. This effort is designed to provide clinicians with important genetic information about how a patient is likely to respond to certain medications. This work is compliant with Mayo Clinic's commitment to safety and quality of practice, while at the same time individualizing patient care. This work is supervised by the Center for Individualized Medicine and the Mayo Pharmacy Formulary Committee, as well as the Mayo Clinic CPC. Pharmacogenomics combines pharmacologic and genomic information, and the Mayo Clinic has started to build alerts about known drug-genome interactions into the enterprise order prescribing tool in Rochester and in other prescribing tools elsewhere in the enterprise. These new alerts are designed to help ensure we prescribe the right drug for the right patient. Specifically, these alerts will identify the relevant genetic test, if one exists, and advise you to either review any previous test results that have been obtained or consider ordering a test as a decision aid. I'll give you some examples. HLA-B5701 genotyping is used when abacavir is prescribed for HIV infections because hypersensitivity reactions are known to be highly associated with the HLA-B5701 allele. These reactions generally occur within the first 6 weeks of treatment and are often nonspecific and include skin rashes, gastrointestinal symptoms, and respiratory symptoms. Fatalities have been reported with abacavir in individuals who carry this allele. Testing for HLA-B5701 genotype and excluding individuals with that genotype from receiving abacavir decreases the incidence of abacavir-related hypersensitivity reactions. Similarly, HLA-B1502 genotyping before the use of carbamazepine for neurological or psychiatric purposes in Asians protects them from another hypersensitivity reaction. Individuals of Asian descent who have this allele are at greatly increased risk of Stevens-Johnson syndrome and toxic epidermal necrolysis. Similar but less well-known associations exist for individuals of Asian descent who are given oxcarbazine, phenytoin, fosphenytoin, and lamotrigine. A third example is the use of thiopurine methyltransferase, or TPMT genotyping or phenotyping to predict the risk of thiopurine toxicity, and that's for drugs such as 5-mercaptopurine or azathioprine. Finally, interleukin 28B, so-called IL-28B genotyping, can be used to predict responses to hepatitis C virus genotype 1 in combination with ribavirin and pegylated interferon therapy. Patients with one genotype will respond more favorably than those with the other genotype, and so when these patients are genotyped, a prediction of their response is offered. These 4 alerts will soon exist in the electronic medical record when individuals go to prescribe medications electronically, and more will be added by the Pharmacogenomic Task Force as it continues its work and identifies additional actionable drug-gene pairs.